Okay, so the topic for today's lab is the cell, right? So one of the first things you have to be aware of is that, all living things are made of cells. If it appears to be an organic molecule or organic in nature, but it's not a cell, then it's not alive. So that's one of the reasons why viruses are not considered to be living organisms because they are not cells. They're just protein packages that require a cell in order for them to replicate and make more copies of themselves. So the cell theory states that the cell is the basic unit of life, and all organisms are composed of cells, and all cells arise from pre-existing cells, right? And uh, like I was saying before, cells are the smallest unit of life. There's nothing smaller than a cell or less complex than a cell that can be considered to be living. All right, and they exhibit self-maintenance and self-replication. So they maintain their systems and they're able to reproduce on their own without the aid of any other cell. Okay, so the main component of the cell that you should be aware of, especially today, is the plasma membrane. All cells are surrounded, or they're bound. The boundary of the cell is what we call a membrane, and the membrane is composed of uh, phospholipids, and I'll talk about that in the next slide. We're 2 slides down. The plasma membrane, this membrane that we're talking about, it's a membrane that maintains the plasma of the cell, the cytoplasm, the liquids, and the organelles is the cytoplasm. So we call it the plasma membrane because it maintains the plasma in one package. The plasma membrane is what we consider to be selectively permeable or semi-permeable. And the reason we say that is because the membrane allows for some things to go through it and others are rejected or are unable to pass through it. Either they can't leave the cell or they can't get into the cell. So, for example, there are molecules that are perfectly capable of going across the plasma membrane. Usually those are small molecules and they don't carry a charge. They're not positive or negative ions, for example. And because they're small, they can diffuse across the plasma membrane. But large or charged molecules are not easily transported across the membrane or are completely unable to pass through the membrane. Okay. Hold on. Okay, so there are 3 ways in which molecules move constantly in and out of the cell. And I do mean constantly because molecules are never standing still unless they're in a state of being frozen, solid. Okay. All right. 2 of the ways are passive. One of those ways is active. And by passive or active, we're talking about what kind of input of energy does it require. Okay, so the 2 passive forms are, well, we just use diffusion, simple diffusion. And diffusion is just the movement of molecules from where they're found in high concentrations to where they're found in lower concentrations. So if they're found wherever it is that they are now, that they're in high concentrations, they want to disperse to where there aren't many of them. So that's passive diffusion. Okay, sorry about that. Interruptions can't be helped sometimes. All right. So you have passive diffusion, which you observe every time you put. Say you can think of a cup of water and a drop of food color. The drop of food coloring is where this molecule that's giving the color is concentrated. It's in high concentrations. You put it into the cup and you don't stir it. Eventually, the entire cup will turn whatever the color of the dye was. It's because the molecules have moved, have diffused on their own until the system that it's in reaches equilibrium. There's also facilitated diffusion, where the molecules that are passing through the plasma membrane are using the laws of diffusion, but they happen to be one of those larger molecules, which don't just easily pass through the membrane. So what happens is that within the membrane, there might be tunnels that we call channels available for these larger molecules to pass through. All right, and those channels might be specifically designed to allow for specific molecules to pass through, or specific groups of molecules, not just any old molecule. Then we have active transport, which requires the input of energy. Right, so the only energy that passive transport uses is ambient energy, the energy from, for example, the temperature of the environment. All right, so with active transport, you need to use ATP. Usually, the only reason you need active transport is because you're moving something against the gradient. What we mean by when I say against the gradient is moving something from where it's found in low concentrations to where it's found in high concentrations. So it's opposite of the direction in which things usually move with diffusion. So this is not diffusion. All right, so energy is needed from the cell. Cell has to produce energy in the form of ATP, and ATP gets broken down in order to release energy. All right, and the molecules that are passing through can only pass through the protein channels. Okay. So here's a picture of what I was talking about. So here you have the plasma membrane, and for cells, it's a double layer, right? You have this is the main component of the plasma membrane, a lipid fatty acid we call phospholipid. Phospholipid. Okay, so here's your phospholipid, and it has a phosphate head. I'm going to write this over here. Phosphate head, which is hydrophilic. What that means is that it can bind and interact with water, and it's got a fatty acid tail, this part here, and that's a hydrophobic tail. Hydrophobic. And what I mean by hydrophobic is that it only interacts with molecules that are not water. It can't interact with water. Right? So this is very similar to what you would find in soap. In fact, it's almost identical. So with soap, how does soap work? Soap takes the fatty acid tail that it's composed of, and it grabs onto other fats that might be on your clothes, the stains on your clothes, or whatever fatty materials you may have on your hands after eating, and it will attach itself on the other end of the molecule to water which is why we use water to wash. All right, this is why soap and water work so well. So since the inside of the cell is aqueous, kind of filled with water, then you have two layers. All right, so here's one layer, and here is another layer, right? So you have phosphate head, fatty acid tail, or phosphate head again. Remember, the fatty acid tails can't interact with water, so the best option they have is to interact with another fatty acid tail. That's why we have bilayer, phospholipid bilayer. We call that bilayer. Two layers of this membrane. Okay, so that membrane is composed of two layers of fatty acids. Phospholipid bilayer right there. Okay, so what I was talking about before is if you have a small molecule, it's not charged, it can go right through, it can pass right through in any direction. But if you have a large molecule, you may have to have a specific place where it can pass through, and that would be the channel protein, or the protein channels. However you want to say it is fine. And note that it's kind of hollow on the inside because it's an empty space. It's a protein that forms a tunnel for the molecules that are much larger than for the large molecules. This tunnel allows them to pass through. There are other protein channels that require the input of energy, so they're not open tunnels, they're kind of closed on one end. So they start off like this. Say you have your lipid bilayer there, okay, your membrane layer, and some little molecule wants to get in, going this way. It'll grab onto it, but then how does it, with the active transport, it will, the proteins will change shape as it passes. As it grabs onto the protein, or to the molecule, I apologize, and then pushes it out, okay, in this direction. Okay, so something approaches, the molecule opens up and changes shape for it to spew it out on the other end. But don't worry too much about that. That's a little bit more complicated than it needs to be for this course. All right, so I'm going to formally define for you guys the term diffusion. And that is the movement of molecules down a concentration gradient, right? So what we mean by down a concentration gradient is that we're talking about the molecule is moving from where it's found in large concentrations to where it's found in little or no concentration. It's absent. The movement of the same kind of molecule from an area where it is found in high concentration to an area where there is less of it, until equilibrium is reached. So by equilibrium, here's a nice picture of equilibrium. So by equilibrium, what we mean is that there's equal distribution of the molecule within the system. The system could be an ocean, a lake, river, pond, puddle, a cup of water, whatever it is that you're talking about. Test tube. Right? So why does diffusion occur? Because of the random movement of molecules that happens all the time. Right? And where are they getting this energy to move around? And that's just from environmental energy. You know, the temperature. As long as the temperature is above absolute freezing, the molecules will be moving. Okay. So like I was saying before, diffusion does not require energy input from the cell. It only requires energy from the environment. Okay, and here's important. When we say that some molecules are able to diffuse across the plasma membrane, we mean that they enter the cell if there is a higher concentration of that molecule outside of the cell. So what I'm trying to say is the molecules will always follow the concentration gradient, right? So if there's more of that molecule inside the cell than outside the cell, the molecule will move away from the cell, will leave the cell. But if conditions are backwards, if there's more of that molecule outside the cell than inside the cell, then the molecule will move into the cell. And then we also have another term, which is osmosis. Osmosis is a special term for diffusion of water across the plasma membrane. So osmosis is just another word for a special kind of diffusion, right? So osmosis is just the transport of water across the plasma membrane of a cell, right? And the water will move into the cell if there is a higher concentration of water outside the cell than there is inside the cell. Okay. So, for example, I don't know, you might get all wrinkly if you take a bath, right? If you take a bath that has no salts in the bath water, and your skin might get a little bumpy or wrinkly because of the extra bumps that you're making because your skin is absorbing water. On the other hand, if you take a little bit too long in the ocean where there's a lot of salt water, you'll also get wrinkly, but this time it's a little bit different, right? You'll feel a little bit softer, and the wrinkliness will be because of a lack of water in your cells. You're kind of allowing water to leave your cells. It's not really that obvious with people, but we're going to do an experiment. We would have done an experiment today that would have shown something very similar, and I'm going to talk about it. Okay. So why do we have to know any of this? All right. Okay. Why do we have to know about diffusion through plasma membranes? Well, this is how you stay alive. All right. Plain and simply, this is how you are alive today and sitting here watching this video. Cells live in environments that can be dangerous or may have food that's available to them outside of the cell, right? Or they have a buildup of excess molecules on the inside of the cell that they need to get rid of. So they need to be able to maintain what we call homeostasis, maintain themselves. And one of the best ways they do this is through transporting molecules across the membrane. Diffusion being the #1 tool. So cells will use diffusion to bring in water, oxygen, to get rid of carbon dioxide. Right? Carbon dioxide is a byproduct of metabolism. Right? They use oxygen, and what comes out is carbon dioxide out of the cells, and then they transport that carbon dioxide. In your bodies, they'll transport that carbon dioxide into the nearest blood vessel. And the blood vessels will take it to the lungs. And then at the side of the lungs, well, what happens at the side of the lungs? You'll have some blood vessels that are rich in carbon dioxide, have high concentrations of carbon dioxide. And guess what happens? The carbon dioxide leaves the blood vessels because they're following the laws of diffusion. And what comes in, when you breathe in, you fill your lungs with air. Well, all that air inside your lungs is rich in oxygen. So what comes into the blood vessels is oxygen, right? And they're transported. That oxygen gets transported, captured by your blood cells, and transported to other parts of your body, So that's one good example of how diffusion is useful for you to stay alive. All right, and the same thing, you eat food, you break down that food into sugar, and so your blood vessels fill up with sugar molecules, and your cells, which have been using sugar, are low in sugar. So now you have a system where blood vessels are full of sugar, so there's high concentrations of sugar in your blood vessels, and your cells are low in sugar because they've been using up whatever they receive. So the sugar will transport itself across plasma membranes from the blood vessels into the cells that need that sugar. They also use facilitated transport, right? Facilitated transport to bring large nutrients, not just sugar, and to take out big amounts of waste, large molecules of waste is what I mean. Okay, active transport can be used to remove dangerous chemicals, say a buildup of salt inside the cell for whatever reason, or you spill a chemical, say you spill a chemical on your hand, as alcohol, for example, or I don't know what, something that is, for example, bleach or something in your house. Well, your cells are going to go into overdrive. Whatever cells are alive after being exposed to this are going to go into overdrive and try to push these molecules out of the cell, even though they're found in higher concentrations outside of the cell. Okay. All right, so there's another topic that we cover in the book, which is tonicity, and tonicity is related to diffusion, but it's related to diffusion in terms of what happens with osmosis. Okay, so we're talking about how water moves in and out of cells according to certain conditions. All right, so cells are often confronted with environments that vary in amount of solutes. That's a dissolved substance in some aqueous environment. The aqueous environment we call the solvent. So usually we're talking about water being the solvent, and the solutes are salts. Right? And so when we ask what is tonicity, we're talking about what is the relative concentration of a salt, the solute, and the solvent, the water. And what is the relative concentration of these things outside the cell compared to the inside of the cell? So you have three possible solutions of tonicity. You have an isotonic solution, you have a hypertonic solution, and a hypotonic solution. Okay, so we're going to talk about that now. Most of the time, cells, in most living things, in people as well, the cytoplasm of a cell, the inside of a cell, has a certain amount of salt. And the concentration of salt in that cell would be about 0.9%. It's very small. It's not absent, but it's very small. All right. And if we confront our bodies or any cell with an environment that has the same or the equal amount of salt in that environment, then you're talking about an isotonic solution. For example, bath salts. If you take a bath in your bathtub, if you're lucky enough to have a bathtub, and you throw bath salts in there, they're going to, I think they're going to bring it up to a concentration of 0.09%. All right? Hopefully, that would be my hope. But say, for example, you get a wound, you're wounded, you go to the hospital because you have this, hopefully this never happens to anyone, but go to the hospital because you have a nasty cut, or you get something in your eyes. They're going to wash your wound or your eyes with not tap water, but they're going to use an isotonic solution. All right, so they're going to wash your wound and wash the cells that have been exposed to the environment with a solution that has an equal amount of salt than the cells inside your body. And they do that because that will reduce the stress of the cells and they'll be able to heal the body faster. Okay, now here we have a picture of some red blood cells that have been exposed to different levels of tonicity. And blood cells, you know, is hollow in the center. Red blood cells do not have nuclei. They've replaced the nuclei with hemoglobin, which allows them to capture lots of oxygen. So a normal red blood cell looks like what we see here in this first picture in square A. Okay, and this blood cell happens to be in an isotonic solution. So the blood cell remains normal. It's not absorbing water, it's not releasing water. It's perfectly happy, it's at equilibrium with the environment. But let's say that we were going to place a cell in an environment where there's a lot of salt, anything above 0.9%. In that case, we would call that a hypertonic solution. Okay, so this over here, this middle picture of a blood cell, this is what a blood cell will start to look like in a hypertonic solution. But why does it look like that? It's all shriveled. It's actually what we call crenated, right? Crenated. This way. It's crenated, shriveled up, because if you place that cell that has 0.9% salt within it into a solution that has, for example, 10% salt, now 0.9 compared to 10, right? That's an order of magnitude more salt than inside the cell. So, and we said before, salt is not a molecule that can transport itself across the plasma membrane easily, because salt ionizes. So you end up with sodium molecules and chlorine molecules, and they're charged molecules. That's what we mean by ionizing. They have a positive and a negative charge, and we said one of the rules for diffusion across the plasma membrane is that molecules should not be charged. So if you can't change the concentration of the salt, what can you change? Well, you can change the concentration of the water. Right? So if you place that cell in a salty environment, a hypertonic solution, then since you can't get rid of the salt inside the cell or take in salt from outside the cell, then the only way to attempt to reach equilibrium is to change the amount of water in the cell. All right, so water is one of those molecules that transports itself across the membrane very easily. So in this case, in a hypertonic solution, water exits the cell. Okay, water exits the cell. However, if we expose the cell to an opposite condition where now the cell has more salt within itself than the environment that it's in, then we're calling that environment a hypotonic solution, right? We're going to put that cell in a glass jar with water that has 0 salt. It's distilled water, for example. Well, in that case, the only way to dilute the amount of salt in the cell so that the system inside the cell and outside the cell is more equal to one another is to absorb extra water. So water moves in. In this case, water moves in. So in the case of a hypertonic solution, water leaves out. In the case of a hypotonic solution, water moves into the cell. And I hope you guys understand why that happens. And in the case of an isotonic solution, there's equal amount of water moving into the cell, there's out of the cell, so the cell maintains it doesn't have an excess number of water molecules, or neither is it lacking sufficient water molecules. Then there's also the topic in this one chapter of learning how to recognize some cells. Right? How do you recognize a prokaryotic versus eukaryotic cell? These are basically the only two types of cells that exist on Earth, right? And the difference is that prokaryotic cells, mainly, they have no membrane-bound nucleus. All right, and eukaryotic cells do. All right, so prokaryotic cells, we're talking about bacteria. They're more primitive, they're simpler, they're smaller. All right, so they do have a plasma membrane, of course, and they may have, I think most bacterial cells, a cell wall as well. But they have no organelles, and like I said before, no membrane-bound nucleus. Okay, so that's important. Your cells, since you are a eukaryotic organism, will have a nucleus that has a membrane surrounding it. It has multiple organelles, and all of those organelles also have a membrane. All right, eukaryotic cells are much bigger as a result, and they have membrane-bound nucleus and membrane-bound organelles. Here's a picture of a bacterial cell. This is a transmission electron microscope graph, and this is just an artist's rendition. Okay, and they have a cell wall, they have a cell membrane just underneath the plasma membrane, but then they do have a nucleus, which is a region where they have their genetic material, but it's not bound by any membrane. Right? Compare that to an animal cell, which is a eukaryotic organism. Here's a nucleus, and it's well defined by that nuclear envelope or nuclear membrane. Okay, so there's a list of stuff for you to remember, right? Prokaryotic cells are small; they're usually less than 10 micrometers in length. Remember a micrometer. If you took a meter stick and divided it 1,000,000 times, that would be 1 micrometer. Each division would be 1 micrometer. So think about how small a bacterial cell is, while a eukaryotic cell is much larger. And the largest single eukaryotic cells out there are large enough for people with really good eyesight to see. Right? We're talking about, for example, an egg cell in humans, for example, or any mammal. Prokaryotic cells are almost always unicellular, and sometimes they're colonial, but colonial doesn't mean multicellular. Colonial means just a bunch of independent cells working together. Eukaryotic cells are multicellular. Example one would be you, right? You're a multicellular organism. Prokaryotic cells, like I said before, no membrane-bound nucleus or membrane-bound organelles. And the eukaryotic cells are the opposite condition. Prokaryotic cells lack a cytoskeleton. The cytoskeleton is like a highway made up of proteins that would allow for things inside the cell to move around in specific directions. Eukaryotic cells are full of cytoskeletons. And when it comes to division, all right, prokaryotic cells will divide by binary fission. All right, what that means is that you have a cell and basically it starts to split and it shares the contents between the two resulting daughter cells. And eukaryotic cells will undergo mitosis, which is division of the nucleus first, and then they undergo division of the cell. Or they'll undergo meiosis, which is like mitosis, but a little bit more complex. What about reproduction? Reproduction is always asexual in prokaryotic cells. So you might stop to ask me, but hey, what about how do they exchange DNA? Well, that can happen. You can have two bacterial cells that will create a little highway between each other, which we call, with little structures called pili, and they'll undergo conjugation. So basically, what that means is that one of them will donate its DNA to the other cell, and then they'll separate. Basically, nothing happens until all of a sudden that one cell starts using all this new DNA. So no new cells result from conjugation, but somehow the cell ends up having new DNA, and eventually, after it undergoes binary fission, you have a number of cells that have different DNA. Now, eukaryotic cells undergo sexual reproduction, and that's, they're taking DNA from two parents and using both sets of DNA and mixing and combining the DNA. And we'll talk more about that next week. And they can also fully capable of just cloning themselves, and that would be asexual reproduction. All right, so that's prokaryotic versus eukaryotic cells. And the other topic in the book is for you guys to get to know plant cells versus animal cells. And be aware right away that plant cells and animal cells are both examples of eukaryotic cells. And there are some blaring differences between plant cells and animal cells. Right away, plant cells have a large central vacuole. And what we mean by that is they have a large bubble inside their cells that contains liquids. That large central vacuole can be a storage for liquid sugar, or sugar dissolved in water, or it can have the toxins that a lot of plants have that keep animals from eating them, anything like that. Just excess water, storage of water. It's very important for plant cells. All right. Animal cells have vacuoles, but they don't have large central vacuoles. They have little ones that roam around the cell. And sometimes those vacuoles are used for digestion. We call those lysosomes. All right, so the contents of those vacuoles vary. Some of those vacuoles are only for carrying food items. Okay. Plant cells have an organ that animal cells do not, and that would be chloroplasts. All right, chloroplasts would be those little green organelles that are responsible for the plant's ability to photosynthesize, to produce sugar with the aid of light. And the one thing that animal cells have that plants don't have is centrioles. And centrioles are part of sexual reproduction. They're used in, and asexual, they're used in mitosis and meiosis. Okay, so plant cells do undergo meiosis and mitosis, but they don't use centrioles. We'll talk more about that next week. And plant cells have a cell wall. Animal cells lack a cell wall. We talked about that yesterday. Okay, so here in this picture, you can see this in your lab manual. Okay, your lab manual should have a diagram of an animal cell and a diagram of a plant cell. And you guys do need to fill these out. So you have the ability to find the answers, to be able to put a name to each one of these letters, to each one of these organelles. All right, so for example, this here is the mitochondria. Mitochondria, which is analogous to a chloroplast. Mitochondria and chloroplasts are little organelles that have their own DNA. Okay, so for example, A here is chloroplast. All right, and you guys can study up on this, and I will post the answers to these tomorrow. Or maybe I'll send them by email. Okay. So here's, I'll fill this one out here. Here's a central vacuole. Large central vacuole. So that's this thing. That's what we were talking about with the plants. Okay. And the animal cell, S are the centrioles. So this one here. Okay, here's a mitochondria. Again, no chloroplasts. And one of the first exercises that you're doing in the lab today would have been to observe a plant cell with easily observable chloroplasts. And that would have been seen with the plant cell from the plant Elodea. Elodea. Elodea or Elodea, however you want to pronounce it, is correct. And that's an aquatic plant. They used to sell these at the local fish stores, the fish shop for aquariums, because people like to grow plants in their aquariums. It's a popular plant for biology because it makes for easy observations. You have a nice plant cell that's rather relatively large, and notice it's empty on the inside. Right? It's empty. And why? Why is it empty? Because it's full of the central vacuole. Right? And then you see all these little green dots. You see all these little green dots around the periphery of the cells. those would be the chloroplasts. And if you were to use the fine focus of the microscope to focus on a different plane, remember back to yesterday's recitation where we were talking about the depth of focus, and you can only focus on a single plane at a time. That is, you can focus on the top of the cell, but you can't focus at the middle of the cell at the same time. So if you wanted to see the middle of the cell, you no longer are able to see the top of the cell. Well, in this case here, you're able to see the top of the cell, but not the inside. And what's surrounding, or what's completely underneath the entire top of the cell, is more chloroplasts. Okay, so it doesn't look empty anymore, only because there's a whole bunch of chloroplasts pressed up against the surface of the cell by that central vacuole. And the chloroplasts are also being pushed to the sides of the cell, pushed up against all the surfaces of that cell. In this slide, I want to talk about one of the other experiments that we would have done. And in this experiment would be diffusion through a plasma membrane. Okay, so diffusion through a plasma membrane. Obviously, we don't have a plasma membrane that's this large, but what we have is something very similar, and it's a dialysis bag. It's a tube, actually. It's a tube of dialysis material. And what's good about the dialysis membrane is that it is also selectively permeable, just like cell membranes. And it selects for things basically the same way that cell membranes do. So small molecules that are uncharged pass through very quickly, but not large molecules or charged molecules. Okay, so for example, in this exercise, I would have been demonstrating this anyway. You guys would not have been doing this experiment. I would have just been demonstrating it for you. In this exercise, I would have cut a piece of tubing of this dialysis membrane. I would have tied it off at one end and on the other end as well, just to make a bag. I would have filled it with glucose, which is sugar, which is a small molecule. So here's a picture of a glucose molecule. It's a simple and small molecule made up of carbon, hydrogen, and oxygen. And it forms this nice little ring because of the bonds between the carbon and oxygen. And it stays at that size. All right. And I also would have filled the bag with starch, which is a much larger molecule. Here's a picture of that, of starch. But note the comparison between glucose and starch. Starch is basically just a chain of glucose. You get rid of this OH, this hydroxide here, and you can attach more. You can attach another oxygen, and to that oxygen, attach it to another carbon, or another carbon ring. This is a carbon ring. This is a carbon ring over here. And you make a chain of carbon rings, and you have starch. So it's basically sugar. It's just a chain of sugars. We call that a polysaccharide. This is a polysaccharide. Right. Okay, so I filled this bag with glucose, and I filled this bag with starch, and I put it in water, and I fill the water with an indicator. And the indicator is just a chemical that's going to alert me to the presence of something else. In this case, the indicator is iodine. Right. You may have had some experience with iodine. They do sell it in pharmacies. But if you say, for example, if you were to put a drop of iodine on your clothes, or especially white T-shirts, or on paper, or anything like on a piece of bread, anything that has lots of starch, it will turn the starch black or purple, dark purple. So what happens is, why did I put iodine there? Well, I know I have starch inside the bag. Why do I need to have an indicator for whether or not I have starch? The idea of the experiment is to show to you guys how large molecules, even though they're similar to small molecules, cannot pass through this membrane. So if the starch were able to leave the membrane, it would fill the cup, and then all of a sudden the color of the cup would be black blue, black purple, because the starch would interact with the iodine in this beaker. But at the end of the experiment, that's not what happened. At the end of the experiment, what we have is a bag that's turned black blue, because the iodine, which happens to be a very small molecule, I2, just two little atoms, two molecules of iodine, which is an element, two little atoms of iodine, which is an element, not molecules of iodine, but two little atoms of iodine, which make one molecule. And that small molecule is able to pass into the bag very easily and stain the glucose on the inside of the bag purple. But we also said that we placed glucose, which is a small uncharged molecule, in the bag. Well, what do you expect to happen there? Well, the glucose is expected to leave the bag, from what we've learned so far. So how do we find out whether the glucose has left the bag? Do we take a taste of the water? Absolutely not. We don't taste anything in labs. Okay, hold on. There's a police officer passing by. Police. Excuse me. Sorry for the noise. And we have another indicator, another chemical process by which we can test for the presence of the glucose in the water outside of this dialysis bag. And that indicator is Benedict solution. Ooh, I made a mistake over here. It's not the presence of starch. I apologize for that. It's the presence of glucose. All right, I have to fix that. Presence of glucose. All right, so Benedict solution is this one, blue. It's a blue caustic chemical. If you were in lab, I'd be telling you make sure you wear gloves, because all of you would have been taking samples and testing it. So you're taking a sample of the water from the beaker and adding it to the Benedict solution, and you'd have to boil it for it to work. Boil it for 5 minutes or so. And the more sugar that is present in that sample, the brighter the color change. So it would go from blue to green to yellow, orange, to dark or bright red, scarlet. Okay, and I can tell you from experience that usually this is what we get. One of these two, depending on depending on how much sugar the instructor puts in the bag at the beginning of the experiment. Okay. So, yes, the glucose does leave the plasma membrane. Okay, it does leave the plasma membrane because it's a small molecule. Okay. Now, there's another experiment where you are observing the effects of tonicity. Right, so we're going to observe some osmosis. And this is an interesting experiment because you're able to conduct this experiment under the microscope as you watch. And you are able to see in real time the changes that take effect. So here is a cell of Elodea, again, Elodea, with under normal circumstances. This is still in its native environment, isotonic. You take a little sample of Elodea with some of the water that it was in, and the Elodea cell looks quite normal. And then when you're making a wet mount, you take a glass slide, you put your specimen in the center of the glass slide, and then you put another glass cover slip on top. So it captures the specimen and whatever water may be in it, maybe along with it. And what happens is, with this experiment, you're able to take a drop of sodium chloride, a solution, water with high concentration of salt, and introduce that salt to the edge of the cover slip. So here is your slide. Okay, and I'll put a leaf here. And you have your cover slip around it. Okay, and you put a little dropper. Let's use this color here. Okay, here's a dropper bottle, and you have sodium chloride inside of it. And you introduce a little drop of that sodium chloride here, and it will diffuse across the cover slip, across your sample, very slowly. And as it does diffuse across the sample, you will see an instantaneous change in the composition of the cells in the Elodea leaf. Okay, so this is your bulb. This is my very bad drawing of the bulb of a dropper bottle, of a dropper syringe. Okay, all right, so as the salt passes across from the edge of the cover slip across your leaf sample, all of a sudden these cells are exposed to a hypertonic solution, right? Remember that. Hypertonic solution, SOLN, period. And what happens to any cell in a hypertonic solution? The cell loses its water, right? The sodium chloride molecules can't go through the plasma membrane, but what can move is the water. So in order for the laws of physics to try to equalize, bring the system to equilibrium and reduce the concentration of salt outside of the cell and increase the concentration of salt inside the cell loses water. And the main source of water for these plant cells is the central vacuole. So the central vacuole shrivels up, and as a result, all of a sudden you see the plasma membrane which would have been attached here. The plasma membrane, you can't see the plasma membrane in this cell because it's attached to the cell wall. But here, it's now over here. It's been detached from the cell wall and is now towards the center. And all the contents of the cell are condensed in the center of this structure. What's left behind is the cell wall. Why does the cell wall not show up? Because the cell wall is a stiff structure. It is not affected as easily by this. Okay? Alright, so this is what you would have observed. You would have seen the cell membrane shrivel up because the central vacuole would have lost its water very, very quickly. There's a second experiment where you would have been able to observe osmosis. This second experiment would have taken a bit longer, though. In this experiment, you would have taken some potato strips, taken a potato, cut out some strips from it, and you would have exposed one of those strips to tap water, and you would expose another one of those strips to salt water using test tubes. You just fill a test tube with tap water, place a strip of potato in that one, and then fill the other test tube with salt water and place the second strip in that one. All right. So, like I was saying before, the salt is not the thing that's going to move across the plasma membrane. It's the water. So the water in tap water, which happens to be hypotonic, what would happen? Think about that. And what would happen with the other one, where the salt water is, which is a hypertonic solution? What would happen to that strip of potato? What would happen to the cells of that potato? All right, and there's a nice video that I found today on YouTube. It's a 5-minute video. Watch that on your own time, and it's basically the exact experiment that we would have conducted in lab, but you guys get to see it as a video rather than just hearing me talk about it. I will give you the results now. All right, the potato that would be in the hypotonic solution, that is the tap water, or distilled water if you want to be really good about this, really scientific about it, use distilled water. That potato will have gained water in an attempt to reduce the concentration of salt to make it more equal to the concentration of salt found outside of the water. So you would have gained turgor pressure. The potato strip would have been turgid, very stiff. You would actually even be able to snap it in half and hear an audible snapping sound, cracking sound. So, you know, you'll be able to observe that the potato, as compared to what it was before, which is a little bit flexible, is now much, much more stiff. However, in the potato strip that got dumped into the salt bath, the hypertonic solution, this is the salt bath, salty test tube. That potato would lose water, right? Because the concentration of salt is higher outside of the cells, so that's going to draw water out. The concentration of water is lower outside of the potato than it is inside the potato. So the water will move where it's found in high concentrations to low concentrations. And as a result, you'll get a limp potato strip. So it'll be floppy, you would be holding it and it would be bending and a little bit harder to snap. You would actually have to just tear it apart to be able to cut it. All right. it would have no turgor pressure at all. All right, so at this point, I want to go into the lab manual itself and go through very quickly. All right, we're going to share my screen. Okay, here we go. All right. All right, so on the first portion of the chapter, it's asking you to examine a prepared slide of bacterium. And it asks you, is there a nucleus visible? So the answer is no, right? I told you guys the bacteria don't have a membrane-bound nucleus, so it's not easy to see that. And if you were to look at some human kidney cells, yes, you would be able to see a nucleus. Now, we saw a nucleus yesterday in the cheek cells, and I showed you a cheek cell picture today as well, and both times you're able to see a nucleus. So the answer would be yes. You can easily answer that. I went over these structures that's in the PowerPoint, so you're able to compare the PowerPoint to this page, and you should be able to fill this out. Okay, the differences between plant cells and animal cells. You can find those differences in the PowerPoint presentation. Now, the animal cell structure, this portion, the plasma membrane, nucleus, all these, yes, please fill this out. Fill this out. All right, this is your homework. You don't have to hand it in to me tomorrow or anything. You hand it in when you hand in the lab manual. So just a quick, easy definition for each one of these structures. You can find this in your lab book. There is a chart that tells you what each one of these is. Also, if you happen to have the lecture textbook, I do believe the answers are there. So you're able to take these diagrams of animal and plant cells and label them, right? So you have a, here, for example, I'll give you another one, nucleolus, there, right? So that's this structure here, nucleolus. And you have the mitochondria, you have the, okay, you have the centrioles, I gave those already. And you do the same thing for the plant cells. So here's your central vacuole, right? Here's your nucleolus, and here's your nucleus, your mitochondria, and your chloroplast. And of course, your cell wall. This might get a little bit more complex with the cell wall. Depends. Right. I think with the other diagram that I showed you was a little bit more complicated. This one's easy, just has a cell wall, so that's this one, cell wall. And this here showing you the cell membrane, which is attached to the cell wall, right? Told you about the chloroplast. Chloroplast here. And I'll give you this one. It's a little bit hard to see. This cytoskeleton. These are little rods here, there. There's another one there, and they're just kind of like highways for the organelles to be able to move around. Okay. Okay, so this was the first observation of the day, which was the Elodea cell structure, and you were able to see the central vacuole and the chloroplasts, these things, that are pushed up against the periphery of the cell. All right, can you locate the cell nucleus? And I'm going to tell you no, because it was very difficult to see that and distinguish it from the chloroplasts. On occasion, some people were able to see, but since we don't stain this cell, it would have been very difficult to see, so I would accept no. Why can't you see other organelles featured in figure 4.3? Okay, so why can't we see any of the other organelles? They are all pushed up against the perimeter, right? The perimeter of the cell. So that's a good answer. All the organelles, including the chloroplasts, are pushed up against the perimeter of the cell, the periphery of the cell. Can you detect movement of chloroplasts in the cell or any other cell? And I was going to tell you yes. And you guys can look up a video of this, but the term we're looking for is cyclosis. And it doesn't always work, but often we are able to see the chloroplasts moving in a clockwise or counterclockwise direction. And they keep moving, that's cyclosis. And because that allows them to take advantage of as much light as possible. So they're, each one of them is taking turns with the light. Here's an experiment that, of course, we're not able to do. But the idea behind this experiment is to show you how diffusion works through different mediums. So we were going to take a petri dish filled with a semi-solid, which is like Jello, but clear. We call it agar. And we were going to put a crystal of potassium permanganate in the center. And what happens is that crystal will diffuse across the gelatin. It would take about an hour for it to really, show you any change, but it would happen. So diffusion through a semi-solid would be slow. Take about an hour or so, but it only moved a couple of millimeters. Then we would compare that to diffusion through a liquid. We would take a petri dish, fill it with water, let the water rest, and then we put a crystal of potassium permanganate in the middle of that water and watch it diffuse. And that would only take about 15 minutes or so. And it would fill the entire petri dish. So you would see that movement through a liquid is much faster. Diffusion through a liquid is much, much faster than it is through a semi-solid. Right? Diffusion through a solid is almost impossible, which is why, we don't bond or diffuse into our tables and our chairs and our floors. Right? Things stay separate. And then we would have compared that to diffusion through air. Here's the problem: diffusion through air, we would be using a perfume, which is not potassium permanganate. So we're introducing another variable, which reduces the value of our experiment. And also, we're not controlling for other variables, like how much air movement is there in the room, and every time somebody breathes, there's an air current being created, or somebody moves around the room, another air current is created. But if we ignore all that, I would have sprayed a little Lysol can in the front of the room, and I would have asked the class to time it and see how long it's for the person all the way at the other end of the room to smell it. And that usually takes about 2 minutes. So the end result is that in which experiment was diffusion the fastest? Well, diffusion, that would be through air, diffusion through air, which is intuitive, right? What accounts for the difference in speed? There's less material for the molecule to pass through, right? There's less friction. Less friction. Less material. Okay. Now, I already explained this experiment. This is the experiment of osmosis. Well, not osmosis, diffusion through a plasma membrane, right? The sugar exits, the iodine goes in, so I2 in, I2 in, C6H12O6, that's sugar. That leaves the dialysis bag. Starch stays put. All right, and the end result has been something that we're going to discuss right here. All right, at the beginning, at the start of the experiment, what is the color of the bag? And because it's full of starch, it would be white. You guys can fill this out and picture this. What about the color of the water in the beaker? Well, because we filled it with iodine, it becomes amber. Okay. At the end of the experiment, though, the color of the bag is now purple. We'll call it purple. That's fine. And the color of the water in the beaker is still amber. But we're going to, so our indicator for starch has worked. It has moved into the plasma membrane, into the dialysis bag, and it has shown that iodine is able to move in. And it also has shown us that starch has not been able to move out of the dialysis bag. So starch can't diffuse out of the bag. But the iodine, I2, is able to go into the bag. It's able to transport itself across the membrane. Now we're going to take, I told you we were going to take a sample of the water from the beaker at the end of the experiment and expose it to the Benedict's test. And what would happen? Well, the Benedict's test would show that the water turns, let's call it red. All right, let's be sure about this in this hypothetical situation. And what does that mean? That means glucose diffused out of the bag and into the beaker. Okay, and you guys can reword that so you can make it fit if you want. Based on the color change noted, what solute diffused the dialysis membrane? All right, from the beaker to the bag, and record your conclusion, which we did. From the results of the Benedict's test on the beaker contents, conclude what solute diffused across. So in that case, it would have been the glucose. In the first case, it would have been the iodine, which diffused. Which solute did not diffuse across the dialysis membrane from the bag to the beaker? And the answer would be starch. How do you know the beaker was full of iodine, right? So if starch had made it through, had diffused, the beaker, sorry, not the beaker, the water in the beaker would have turned purple. Excuse me. Okay, this experiment, we can cross this out. We never do this experiment in the lab. Okay, so this osmosis demonstration, we don't do, so we can cross that out. All right, and then we're going to talk about tonicity in cells. Tonicity is the relative concentration of solute particles, and therefore also of solvent water, outside the cell compared to within cell. An isotonic solution has the same concentration of solute as the cell. When cells are placed in an isotonic solution, no, they're not saying that there's no movement of water. They're saying there's movement in the amount of water in out of the cell, right? So we'll always move across the plasma membrane. It's just that in an isotonic solution, there is no difference in the amount that's going into the amount that's a solution has higher solute, therefore lower water than the cell. When cells are placed in a hypertonic solution, water moves out of the cell into the solution. Okay, so that takes in a hypotonic solution, a hypotonic has a lower solute, therefore more water concentrated in the cell. When cells are placed in a hypotonic solution, water moves from the solution into the cell. Right? So that's the case in this picture. This is the isotonic here. Isotonic. They're all labeled here. All right. Eventually, what happens to this is this cell will burst. And the term we use for this is lysis. All right. So it'll undergo hemolysis. If we're talking about a blood cell, we call it hemolysis. Hemolysis. All right. Now, obviously, we can't do this experiment. We're not going to be able to do the feasibility experiment. We can do the conclusions at the end. All right. And we were going to do the experiment of adding salt to plant cells, the Elodia. I explained this in the PowerPoint. All right. And, when plant cells are in a hypotonic solution, such as fresh water, the large central vacuole gains water and exerts pressure called turgor pressure. The cytoplasm, including the chloroplasts, is pushed up against the cell wall. When plant cells are in a hypertonic solution, such as sodium chloride, 10%, the central vacuole loses water and the cytoplasm, including the chloroplasts, pulls water from the cell. This is called plasmolysis. All right, so hemolysis is plasmolysis, but it's in terms of a blood cell. Plasmolysis is what is the term used for all cells. Whenever they burst because either they lost their water or they gained too much water and exploded. All right, so we can fill this out. Appearance of the cells in the hypotonic solution would have been normal. And I guess you can say it's turgid. The term you're looking for is turgid. In a hypertonic solution, okay, they would appear shriveled up. All right, so the cell membrane is kind of ruptured a bit, so you call this plasmolysis. Then they're talking here about the experiment with the potato strips, which you guys can watch a 5-minute video on. And all right, so you place one potato strip in tap water or some sort of hypotonic solution, like tap water or distilled water, and the other one in the hypertonic solution, such as salt water. Which tube has the limp potato strip? And that would be the hypotonic solution. Just write hypotonic solution. All right, because use tonicity to explain why water diffused out of the potato, because it was you guys use full sentences, right? Because it is in a hypotonic solution. And water. Sorry, not hypertonic. Ooh. I apologize for that. I was confusing myself here. This be hypertonic. And water leaves the cells. Which tube has a stiff potato? And that would be the hypotonic. And of course, again, here you use better sentences than I do, please. Use full sentences. Because it is in a hypotonic solution. And what happens to the water? Water enters the cell. So the results, conclusions are, in a hypotonic solution, animal cells will gain water, H2O. In red blood cells, this is called hemolysis. In a hypertonic solution, animal cells lose water. In red blood cells, this is called crenation. In a hypotonic solution, the central vacuole of Elodea exerts turgor pressure. Turgur. And chloroplasts are seen, they're pushed up at the perimeter, right? Pushed up at the perimeter. Perimeter, okay? All right. In a hypertonic solution, the central vacuole loses water, and they said plasmolysis occurs. The cytoplasm plus the chloroplasts are seen in the center. In a hypotonic solution, potato strips hypotonic, they've gained water and become turgid or stiff. In a hypertonic solution, potato strips lose water and become limp. Okay. All right. The pH of the cells we are not going to discuss today. We're going to do pH. We're going to discuss pH tomorrow, but we're not going to cover it in terms of this chapter. Do not fill this out. You don't have to. We're not going to do this at all. You don't do this. Okay. We don't do this. And that brings us to the end of the chapter. All right. What regulates the movement of molecules into and out of the cytoplasm? Well, they're asking what is it that stops things from getting in and helps things get out? And that's the plasma membrane. What kinds of cells lack a nucleus? Prokaryotic. What structures Uh-oh. I'm losing battery, and I don't know why. Okay, now I fixed the battery issue. What structures associated with prokaryotic and eukaryotic cells carry out protein synthesis? I didn't go over that, but I'll give you the answer. Ribosomes. Ribosomes, there you go. What two things are found in the nucleus of a eukaryotic cell? So one of the things is DNA. Okay, and they also have a central organelle, which is the nucleolus. That's fine. You can just say nucleolus and DNA, or genetic material. What plant cell organelle carries out photosynthesis? I think we all know that's the chloroplast. What molecule is associated with plant cell walls? And that would be cellulose. Cellulose. I'm going to give you the answer to that one. Cellulose, which happens to be extremely difficult to digest, which is why they tell you to eat vegetables. You can't really digest them, not the cells themselves, which becomes roughage and helps your digestive system. Termites are known for eating wood, which is cellulose, but did you know that it's not the termite that's able to digest the wood? The termite has a symbiotic, single-celled organism, a protist, like Euglena from yesterday, that helps the termite digest the wood. But the story doesn't end there. Even this protist, this single-celled organism, is unable to digest cellulose. The protist itself has another endosymbiotic organism within its cell, and those are bacterial cells. And it's these bacterial cells that are able to digest cellulose. So it's quite a difficult thing to digest this cellulose. And in nature, things that you can see that can digest cellulose would be fungi. Whenever you see a fungus growing on a tree, that's what it's doing. It's digesting wood, and it happens very slowly. So cellulose happens to be an excellent material if you want to make sure that it doesn't get broken down very easily. What energy ATP producing organelle is found in animal and plant cells? Okay, so that would be the mitochondria. Mitochondria. What is the movement of molecules from an area of higher to lower concentration called? Diffusion. I hope that I eliminated any confusion about the diffusion that you may have. What is the movement of water across the plasma membrane called? So this is a specific type of diffusion, and that is osmosis. Does water move into or out of the cell? Out of the cells, they're placed in a hypotonic solution. Hypotonic solution being a solution that has less salt than what's present inside the cell. Therefore, water moves in. What kind of solution causes coagulation to happen to red blood cells? That would be hypertonic. What kind of solution causes chloroplasts to be pushed outwards against the cell wall? That would be hypotonic or isotonic. Let's go with isotonic because that's the normal condition. And the problem with hypotonic is that hypotonic may actually cause the cell to burst. If a solution has more hydrogen, although it doesn't really happen with plants bursting like that because of the cell walls. So technically you would be correct if you wrote isotonic or hypotonic, but let's go with isotonic. 13. If a solution has more hydrogen ions than hydroxide ions, this is acidic or basic. So now it's acidic, but we're going to cover it tomorrow. All right. What is present in human blood that ensures blood pH is maintained at about 7.4? We don't need to know this. Okay. What cellular components are common to prokaryotic, eukaryotic cells? Well, they both have a cell membrane, right? Bacterial cells and plant cells and fungal cells all have cell walls, so you can technically say cell walls, even though animal cells don't have cell walls. We can broaden our minds and include, plant cells and fungal cells do have cell walls as well as the bacteria, and many protist cells also have cell walls. And ribosomes is another thing. Ribosomes, all cells have to produce proteins, and ribosomes are essential for that. Let's see, ovarian follicle cells produce estrogen, which is a steroid. What organelle will be present in abundance so that the follicle cells might perform this function? The answer would be a vacuole, which stores hormones. You can put that into a nice sentence. Contact lens solution is described as a sterile, buffered, isotonic aqueous solution. Explain the importance of the adjectives buffered and isotonic. All right, so why is it important to be isotonic? All right, because it has the same salt solution as the cells in your eyes. This is a good question. In your eyes. Imagine if your contact lenses were dipped in a hypotonic solution and suddenly you put that hypotonic solution on your eyes. It would sting because all of a sudden water wants to get into your eye cells. Or if it was in a salty solution, extremely salty solution, then all of a sudden your eye will shrivel up, and that would not be good, of course. And buffered. What is a buffered? What is a buffer? We actually discussed this more so tomorrow, but I'll tell you anyway. A buffered solution contains a buffer, obviously, but the buffer maintains a stable pH, which is important for the function of proteins. And all cells use proteins to function, especially your eyes. So if you have your contact lenses in that buffered solution, it's not going to change the pH of your eyes. You don't want to put an acidic solution on your eyes or even a basic solution. You want to put a nice neutral solution on your eyes so that everything stays functioning. Oh, and that would be the end. All right. I think we did well. I'm going to stop screen sharing now and open up the chat in case you have any questions. Number 16. Are you talking about the PowerPoint slide or are you talking about the chapter? In the laboratory review. Okay. Let me screen share again. Number 16. Okay. Let me get to that. Okay, so ovarian follicle cells produce estrogen, which is a steroid. What organelle will be present in abundance so that the follicle cells might perform this function? I would say the vacuole, all right, which stores chemicals or hormones, what the estrogen is. So the cell has to be able to store these chemicals somewhere, and the vacuoles would be the best place for that. Okay, I'm gonna cut off the recording now.